last video we looked at some basic points about Bacillus anthracis. In this video we want to cover lab diagnosis of anthrax. This is important, so please pay attention here. Specimen. First of all, what specimen do you collect? Specimen. What specimen do you collect for anthrax? Because it will give you a malignant pustule. So you have to collect pus. Sputum if it is pulmonary anthrax, and if it is intestinal anthrax you can collect blood, and you can also collect CSF. Okay. Is this much clear? I hope you remember the last video. We have told you the types of anthrax, right? Cutaneous, pulmonary, intestinal. So based on the type of anthrax you can collect the specimen. Very good. Now move on. As soon as you take the specimen, you will do direct microscopy, direct demonstration of the bacteria. Under direct demonstration, you have gram staining. This will be what you already know. Gram positive, rectangular rods or rectangular bacilli. You remember the last diagram, right? We have shown you this one. Gram positive rectangular bacilli. The spores are there which are not bulging. That's why it looks like a straight line. Oval spores are there. You can see them, right? Very good. Under direct demonstration, you have McFadyen's reaction. Some people may say McFadyen reaction, but actually it's McFadyen reaction. Because you have to write the spelling in the exam, I am telling you the correct way of telling it. McFadyen. McFadyen reaction. It is a direct demonstration. This McFadyen's reaction, you will actually use a stain which is polychrome methylene blue stain. And once you use the stain on the anthrax bacteria, you will see purple capsule which surrounds the blue bacilli. Let me show you the diagram here from Apurva Shastri. So here you can see the first diagram A, that is amorphous purple capsule surrounding blue bacilli, polychrome methylene blue stain. This is McFadyen reaction. The first one here. This is McFadyen reaction. If you draw this diagram, you will get good marks. McFadyen reaction, right? Let's continue with the direct demonstration after McFadyen reaction. According to the spelling, I will tell you McFadyen reaction. After that, you have direct immunofluorescence. Okay, you will detect capsular antigen in that. Then you have Ascoli's thermoprecipitation test. Sometimes they will put this Ascoli thermoprecipitation test even under serology. Anyways, you can write it here or there. Anthrax. See, Ascoli's thermoprecipitation test is a ring precipitation test. Okay, when you are using a non-viable bacilli, you have what you will do. You will take the bacilli. Okay, boil them, filter them, everything. Antigenic extract, you will layer it over anthrax antiserum in a narrow capillary tube. So you have the antibodies here. Then here you are putting from top, what are you putting? The antigen. Fine, that's all. Down there is antibody. That is anthrax antiserum you have here. On top you are putting the antigen. And in the middle a ring precipitate is formed. This is very, very simple test, okay? Nothing complicated. And if you draw diagram, it's even better. You can do this test for many things, okay? Many bacteria you can do this test. So we have covered direct demonstration. Let us move on to culture. Culture you can do on nutrient agar. Nutrient agar. Medusa head appearance. Medusa head appearance colonies. The colonies will have an appearance of medusa head. Let me show that to you. This one. like snakes coming from her head. You remember that Greek goddess or something? Medusa head. Colonies of bacilli anthracis on nutrient agar. Nice, no? That will come in culture. Then you have blood agar. Blood agar you will have dry, wrinkled, non-hemolytic colonies. They are non-hemolytic. Please note, they are non-hemolytic. They didn't do any work. Blood agar. Look at this one. Non-hemolytic, dry, wrinkled colonies of Bacillus anthracis on blood agar. Can you see how they are? They're looking like snowflakes almost. Moving on. In colony culture anthrax, you will put in nutrient agar. You will put it on blood agar. Then you will put it on gelatin stab agar. Okay. Gelatin stab agar. Inverted filtrate appearance. Inverted filtrate appearance. Please note that. I will show that to you. Here can you see? Gelatin stab agar, inverted filtrate appearance. Then you have some selective media. Selective media, you have solid medium with penicillin, string of pearl colonies. Then you have the pellet media. Let me show you what diagrams are available online. Okay. Look at this one. I got it online from SlideShare. So here you can see string of pearl appearance with penicillin. Not able to see much actually. It differentiates something. Okay. It differentiates anthrax and Bacillus cereus. Let's move on to the PLET medium. PLET medium. What information do we have about PLET medium? Now, PLET medium is nothing but polymyxin, lysozyme, EDTA and thallus acetate added in heart infusion agar. So, you will take heart infusion agar. To it, you will add polymyxin, lysozyme, EDTA and thallus acetate. Okay, acetate. So you can isolate Bacillus anthracis from mixtures of other spore-bearing bacilli. If you use PLET medium, anthrax, say PLET anthrax, solid medium with penicillin, anthrax, string of pearls. It's not so easy, right? Nutrient agar, Medusa head appearance, blood agar, wrinkled, dry wrinkled, gelatin stab agar, inverted filtrate appearance. Selective media, you have solid medium with penicillin and PLET media. Solid medium with penicillin, it will give string of pearls, and PLET media, it will contain polymyxin, lysozyme, EDTA, thallus acetate. You can use it to isolate from other spore-bearing bacilli. Revision slide, please. Revision. So far what we have seen, actually there is more. We will continue in the next video. So far what we have learned: specimen collection, direct microscopy. In direct microscopy you saw gram stain. Then you saw McFadden reaction. Then you saw some Ascoli's ring precipitation test. You saw direct immunofluorescence. Ascoli's, what was that? Thermoprecipitation test. Direct immunofluorescence, McFadden reaction, where you saw what? Blue bacilli surrounded by purple capsule. You can draw the diagram to get more marks. Okay. McFadden reaction. Ascoli's thermoprecipitation test. Then we saw culture. Culture, you saw nutrient agar. In nutrient agar, what can you actually see? In nutrient agar, you can see medusa head appearance, right? Appearance. Then in blood agar, you can see dry, wrinkled, non-hemolytic colonies. Gelatin stab agar, you can see inverted fir-shaped appearance. Then you have some selective media like solid media with penicillin. There you will see string of pearl colonies. String of pearls, please don't forget string of pearls colonies. Then string of pearls. Then last one you remember, plet, right? P-L-E-T. Plet media, that is nothing but polymyxin, lysozyme, EDTA, thallus acetate. Added in heart infusion agar, you can isolate the Bacillus anthracis from other spore-bearing bacilli. Just small things before I close this video. For all lab diagnosis, you should write PCR, ELISA, write them separate only, okay? So you can fill up a lot. PCR, ELISA, all this you write, okay? Also, you can write molecular typing, antibiotic sensitivity testing. Okay, standard things you write for lab diagnosis, and also write that before giving antibiotics you should collect the specimen and be very careful with the samples because they are highly infectious. These are standard things that you should write for all lab diagnosis, okay? Now, the thing with anthrax is it has so many keywords in the lab diagnosis, right? McFadden, ascolith, medusa head appearance, gelatin stab, inverted fir, string of pearls, too many points. So that's why they like to ask this question, okay?